Picture this: a barren rock hurtling through space, its surface a molten hellscape of fire and fury. Asteroids rain down like cosmic hail, each impact releasing energy enough to vaporize oceans. The sky glows red with volcanic rage. No birds sing, no trees rustle, no bacteria crawl. Nothing lives here. Nothing has ever lived here. This is Earth, 4.5 billion years ago, and yet somehow, impossibly, within just a few 100 million years, life will emerge from this chaos. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Please like the video, subscribe for more like this, and comment below with where you're checking in from. Tonight, we're embarking on perhaps the greatest detective story in all of science. We're going back to the very beginning, to that mysterious moment when lifeless chemistry transformed into living biology. We'll walk through ancient oceans that don't exist anymore, peer into volcanic vents miles beneath primordial seas, and witness the first fragile membranes that separated self from not self. We'll meet molecules that learned to copy themselves, tiny compartments that grew and divided, and the last common ancestor from whom every living thing on Earth descended. This is not a simple story. It's not a story with easy answers or clear heroes. It's a story written in chemical signatures, trapped in ancient rocks, in the genetic code shared by every organism alive today, in laboratory experiments that recreate conditions from before time itself had much meaning. It's a story that scientists have been piecing together for decades, and one where new discoveries continue to reshape what we thought we knew. So, settle in, let your breathing slow. We're about to journey back to the very dawn of existence to understand how, against astronomical odds, the universe learned to know itself through us. Chapter 1: A Planet Born in Fire. Let's begin at the very beginning, 4.5 billion years ago. Give or take a few tens of millions of years, there was no Earth. There was only a swirling disk of dust and gas orbiting a young star. This protoplanetary disk was the leftover debris from the Sun's formation, a cosmic construction site where planets would eventually take shape. Within this chaos, gravity was at work. Tiny particles collided and stuck together. Dust grains became pebbles. Pebbles became boulders. Boulders became planetesimals, objects hundreds of kilometers across. And these planetesimals, in turn, collided with catastrophic violence, building ever larger bodies through a process called accretion. One of these growing worlds would become Earth, but its birth was anything but peaceful. The infant Earth was a ball of molten rock. Every collision added more mass and generated tremendous heat. The kinetic energy of impact converted directly into thermal energy, enough to keep the entire surface liquid. Add to this the heat from radioactive decay, which was far more intense in those early days when short-lived radioactive elements hadn't yet decayed away. And you have a planet that was essentially a magma ocean floating in space. The temperature at the surface may have exceeded 1500 degrees Celsius. There were no continents, no ocean floors, just endless churning lava. Heavier elements like iron and nickel, drawn by gravity, sank toward the center, beginning the process of differentiation that would create Earth's iron-rich core. Lighter elements rose toward the surface, where they would eventually form the mantle and crust. Above this molten surface, an atmosphere began to form, but it was nothing like the air we breathe. The earliest atmosphere was likely dominated by hydrogen and helium, the lightest elements captured from the solar nebula. But Earth's gravity wasn't strong enough to hold onto these gases for long. They leaked away into space, molecule by molecule, leaving behind a vacuum. Then came volcanic outgassing. As the interior of the planet continued to heat and churn, volcanoes began releasing gases trapped in the rock. Water vapor, carbon dioxide, nitrogen, methane, ammonia, and sulfur compounds billowed forth in enormous quantities. This secondary atmosphere was thick and hot, dominated by carbon dioxide and water vapor, creating a powerful greenhouse effect that kept surface temperatures brutally high. But the most dramatic event in Earth's early history was yet to come. Somewhere in the young solar system, another planetary body was forming. Scientists call it Theia, after the Greek Titaness, who was mother of the moon goddess Selene. Theia was roughly the size of Mars, perhaps 10% of Earth's mass, and its orbit was converging with Earth's. The collision, when it came, was the most violent event in our planet's history. The exact nature of this impact remains debated. Some models suggest a glancing blow, with Theia sideswiping Earth at an oblique angle. Others propose a more head-on collision, but the result was the same: catastrophic destruction and transformation. The two bodies merged, their mantles mixing in a spray of vaporized rock. Much of Theia's iron core sank into Earth's core, adding to its mass. But a significant amount of material, primarily from the mantles of both bodies, was ejected into orbit around Earth. This debris disk, glowing white-hot and composed of molten rock and vapor, quickly coalesced under its own gravity. Within perhaps just a few tens of thousands of years, the Moon was born. The impact and its aftermath left Earth fundamentally changed. The planet's rotation was faster, with days lasting perhaps just 5 hours. The collision had added angular momentum to the system, though gravitational interactions with the Moon would gradually slow Earth's rotation over billions of years. The impact also likely tilted Earth's rotational axis, giving our planet the seasons we experience today. More importantly for our story, the moon-forming impact essentially reset Earth's geological clock. Any crust that had begun to form was obliterated. The entire surface returned to a molten state. If any chemistry relevant to life's origin had been occurring before the impact, it was erased. Life on Earth could only begin after this cosmic catastrophe. Following the impact, Earth slowly began to cool. The surface, exposed to the cold vacuum of space, radiated heat away faster than the interior could supply it. A thin crust began to form, like skin on cooling pudding. But this early crust was unstable. Convection currents in the mantle constantly broke it apart and dragged it back down into the molten interior, only for new crust to form and the cycle to repeat. The atmosphere, now enriched with gases from both the impact and ongoing volcanism, was thick with water vapor. As the surface temperature gradually dropped below the boiling point of water, something remarkable happened. It began to rain. Not just rain, really. More like a deluge that lasted for millions of years. Water vapor condensed out of the atmosphere in torrents, falling onto the hot surface where it immediately flashed back to steam, only to rise, cool, and fall again. Slowly, gradually, as the crust cooled further, the water began to collect. Puddles formed in low-lying areas. Puddles became pools. Pools became lakes. And eventually, over millions of years, a global ocean covered most of the planet. by about 4.4 billion years ago, just 100 million years or so after the moon-forming impact, Earth had liquid water on its surface. This is evidenced by tiny crystals of the mineral zircon found in Western Australia, which contain chemical signatures consistent with formation in the presence of liquid water. These zircons are among the oldest materials we've found on Earth, survivors of an almost incomprehensible span of time. But Earth's trials weren't over. The inner solar system was still a shooting gallery. For hundreds of millions of years, asteroids and comets continued to pound the young planets. Some of these impacts were large enough to boil away portions of the ocean, creating vast clouds of steam. Others may have delivered important volatiles, including more water and organic compounds. This period of intense bombardment, sometimes called the Late Heavy Bombardment, didn't fully end until around 3.8 billion years ago. Only then did Earth finally settle into a more stable configuration, with impacts becoming less frequent and less severe. By this time, perhaps 4.3 to 4.2 billion years ago, conditions on Earth had become potentially suitable for life. The surface had cooled to temperatures where liquid water could exist stably. The atmosphere, while still very different from today's, was no longer unbearably hostile. Volcanic activity remained intense, creating hydrothermal systems both on land and in the oceans. Chemical reactions were occurring constantly, driven by energy from lightning, ultraviolet radiation from the young sun, and heat from Earth's interior. The stage was set. The ingredients were present. The window of opportunity had opened. Somewhere on this water-covered world, in ways we're still trying to understand, chemistry was about to cross the threshold into biology. Chapter 2: The Warm Little Pond and the Primordial Soup. The question of how life began has fascinated humans for as long as we've been able to contemplate our origins. Ancient cultures had their creation myths. Many religions offered divine explanations. But as science developed, particularly in the 19th and 20th centuries, thinkers began to search for natural mechanisms. One of the most famous early speculations came from Charles Darwin himself. In an 1871 letter to his friend, the botanist Joseph Hooker, Darwin mused about life's origins in what he called a warm little pond. He imagined a body of water containing ammonia, phosphoric salts, light, heat, and electricity, where a protein compound might be chemically formed. This protein, Darwin suggested, might be ready to undergo still more complex changes, ultimately leading to the first living organism. Darwin's idea was prescient. He understood that life must have emerged through natural processes from non-living materials, and he correctly intuited that this would require the right chemical ingredients, a suitable environment, and sources of energy. What Darwin couldn't have known was just how complex this process would turn out to be, or how long it would take science to uncover even partial answers. The modern scientific investigation into life's origins really began in the 1920s with the independent work of two scientists, Alexander Oparin, a Russian biochemist, and J.B.S. Haldane, a British geneticist. Both proposed remarkably similar theories about how life could have emerged on the early Earth. Oparin published his ideas in 1924 in a pamphlet called The Origin of Life. He proposed that Earth's early atmosphere was very different from today's, lacking oxygen but rich in hydrogen, methane, ammonia, and water vapor. This reducing atmosphere, energized by ultraviolet light from the sun, could have driven chemical reactions that produced simple organic molecules. These molecules would have accumulated in Earth's oceans over vast periods of time, creating what Oparin called a primordial soup. Within this soup, Oparin suggested, molecules could have continued to interact and combine, growing more complex through natural chemical processes. Eventually, he proposed, some molecules formed droplets or cosovates that could absorb other molecules and grow, a precursor to true cellular life. Haldane published similar ideas in 1929. He too envisioned a reducing atmosphere and the synthesis of organic compounds powered by ultraviolet radiation. He famously wrote that the primitive oceans must have reached the consistency of hot, dilute soup. Like Oparin, Haldane proposed that increasingly complex molecules would form in this soup until eventually something arose that could reproduce itself. Together, their work became known as the Oparin-Haldane hypothesis, or the primordial soup theory. It was an elegant idea. Life emerged gradually from simple chemistry, with organic molecules building up in concentration over millions of years until conditions were right for the first living cells to appear. For decades, the primordial soup theory remained just that, a theory. There was no way to test whether the proposed chemical reactions could actually occur. Then, in 1953, came a breakthrough that would capture the world's imagination. Stanley Miller was a graduate student at the University of Chicago, working under the Nobel laureate Harold Urey. Miller designed an experiment to test whether organic compounds could form under conditions thought to resemble the early Earth. He created a closed system containing water, methane, ammonia, and hydrogen. He heated the water to create vapor, circulated the gases through the system, and used electrical sparks to simulate lightning. The experiment ran for a week. When Miller analyzed the results, he was astonished. The water had turned a reddish-brown color. Chemical analysis revealed that 10 to 15% of the carbon from the methane had been converted to organic compounds. Most remarkably, several amino acids had formed, including glycine and alanine, which are fundamental building blocks of proteins in all living organisms. The Miller-Urey experiment, as it came to be known, was headline news. Here was laboratory proof that life's building blocks could form naturally from simple starting materials. It seemed to validate the primordial soup hypothesis and suggested that the origin of life might be more straightforward than anyone had dared hope. Over the following decades, variations of the Miller-Urey experiment produced even more impressive results. Scientists created all 20 amino acids found in proteins, along with sugars, lipids, and even the nucleotide bases that make up DNA and RNA. It appeared that given the right conditions, the chemistry of life emerged almost inevitably. But as appealing as the primordial soup theory was, it faced serious challenges. First, there was the question of Earth's early atmosphere. Modern geological evidence suggests that the atmosphere was not as reducing as Oparin and Haldane had assumed. Instead of being rich in hydrogen and methane, it probably contained more carbon dioxide and nitrogen. This matters because the Miller-Urey experiment works much less efficiently in a less reducing atmosphere. Some organic molecules can still form, but not in the same abundance or diversity. Second, there was the concentration problem. Even if organic molecules formed in the oceans, they would have been incredibly dilute, dissolved in an enormous volume of water. For these molecules to interact and form more complex structures, they need to be concentrated. In a vast ocean, this seems unlikely. The molecules would be too far apart, too diluted to bump into each other frequently enough for interesting chemistry to happen. Third, and perhaps most fundamentally, there was the energy problem. The primordial soup theory never really explained how the first living cells obtained energy. Modern life harnesses energy through complex biochemical pathways, but these pathways require sophisticated proteins and cellular machinery. How could the first cells, lacking all this machinery, power the chemical reactions necessary for life? These objections didn't kill the primordial soup theory, but they did lead many scientists to search for alternative explanations. And in the late 20th century, a new idea began to gain traction, one that would shift the focus from Darwin's warm little pond to some of the most extreme environments on Earth: deep-sea hydrothermal vents. Chapter 3: The Deep-Sea Garden of Eden. In 1977, a team of geologists was exploring the Pacific Ocean floor near the Galapagos Islands, using the deep-sea submersible Alvin. They were studying the mid-ocean ridge, the underwater mountain chain, where Earth's tectonic plates pull apart and new ocean crust forms. What they discovered would revolutionize biology. At depths of 2,500 meters, in complete darkness and under crushing pressure, they found something nobody expected: an oasis of life. Tall chimneys of mineral deposits rose from the seafloor, billowing forth dark, superheated water. Around these chimneys, life flourished. Giant tube worms, some over 2 meters long, swayed in the currents. Blind shrimp crawled over the mineral formations. Mussels and clams clustered thickly around the vents. White bacterial mats covered everything. These were the first deep-sea hydrothermal vents ever observed, and they would come to be called black smokers because of the dark plumes they emitted. The water gushing from these vents was hot, sometimes exceeding 400 degrees Celsius, laden with dissolved metals and sulfur compounds. By all conventional wisdom, nothing should have been able to live there. Yet, not only did life exist at these vents, it thrived. Even more remarkably, this ecosystem didn't rely on photosynthesis. On the surface, nearly all life depends on energy from the sun, captured by plants and other photosynthetic organisms. But at these deep-sea vents, miles below the reach of any sunlight, life had found a completely different energy source. The key was chemosynthesis. Bacteria living at the vents used chemical energy from the vent fluids, particularly hydrogen sulfide and other reduced compounds, to produce organic molecules. These bacteria formed the base of the food chain, supporting all the other organisms in the ecosystem. The giant tube worms had no mouth and no digestive system. They survived entirely through a symbiotic relationship with chemosynthetic bacteria living in their tissues. The discovery of these black smoke vents immediately sparked speculation about life's origins. If life could exist in such extreme conditions today, powered by geochemical energy rather than sunlight, perhaps life originally emerged in similar environments on the early Earth. But black smokers posed a problem for origin of life theories. They were too hot. At 400 degrees Celsius, complex organic molecules break apart. Even the hardy bacteria that live at these vents don't thrive in the superheated vent fluids themselves. They live in the cooler mixing zones where hot vent water meets cold ocean water. It seemed unlikely that the delicate chemistry required for life's origin could occur in such extreme heat. Then, In 2000, another type of hydrothermal vent was discovered. Called the Lost City, this vent field in the mid-Atlantic Ocean was fundamentally different from the black smokers. Instead of the scalding temperatures of magma-heated water, the Lost City vents were warm but not hot, with temperatures ranging from about 40 to 90 degrees Celsius. Instead of being acidic and metal-rich, the vent fluids were alkaline, with a pH above 10. Most importantly, these alkaline vents were powered by a different geological process called serpentinization. When seawater infiltrates into the rocky ocean crust and reacts with minerals like olivine and pyroxene, the water chemically modifies the rock, producing new minerals, including serpentine. This reaction generates heat, produces hydrogen gas, and creates strongly alkaline fluids. The Lost City and other alkaline hydrothermal systems immediately became the leading candidates for life's birthplace. They offered several key advantages over the primordial soup scenario. First, they provided structure. The vent chimneys are riddled with tiny pores and channels, creating countless microenvironments separated by thin mineral walls. These natural compartments could concentrate organic molecules and keep them from being diluted in the vast ocean. They provided a physical framework that could support increasingly complex chemistry. Second, they provided the right chemistry. The alkaline, hydrogen-rich vent fluids mixing with the more acidic, carbon dioxide-rich ocean water created chemical gradients across the mineral barriers. These gradients, particularly the difference in pH, represented a source of free energy that could drive chemical reactions. Third, and most crucially, they provided a solution to the energy problem. The mineral walls of the vents, particularly those rich in iron sulfide, could act as natural catalysts, and the pH gradient across these walls, with alkaline fluid on one side and more acidic water on the other, creates what's called a proton gradient, a difference in the concentration of hydrogen ions. This matters because proton gradients are exactly what modern cells use to generate energy. In every cell in your body right now, structures called mitochondria are using proton gradients to produce ATP, the universal energy currency of life. The protein complexes that do this, ATP synthase, and the electron transport chain, are found in virtually all living organisms, suggesting they trace back to very ancient times. Could it be that life didn't invent this energy generation system from scratch? Could it be that the first living cells emerged in an environment that naturally provided the kind of proton gradient they would eventually learn to create for themselves? This idea, developed most fully by the geochemist Michael Russell and his colleagues starting in the early 1990s, suggests that alkaline hydrothermal vents served as natural electrochemical reactors. The micropores in the vent structures, bounded by thin iron sulfide membranes and transected by natural proton gradients, provided ideal conditions for the kind of chemistry that could lead to life. Recent experiments have started to support this hypothesis. Researchers have built laboratory reactors that mimic alkaline vent conditions, with alkaline fluid on one side of an iron sulfide membrane and acidic fluid on the other. In these reactors, simple carbon compounds like formaldehyde can be synthesized from carbon dioxide and hydrogen. While the yields are still low, they demonstrate that the basic chemistry is possible. Other experiments have shown that the kinds of simple fatty acid vesicles that might have formed the first cell membranes are actually more stable in the warm, alkaline, salty conditions of hydrothermal vents than in the cooler, fresher conditions of Darwin's warm little pond. The harsh environment that seemed so hostile to life's origin might actually have been ideal. The hydrothermal vent hypothesis also helps explain something puzzling about biochemistry. If you look at the metabolic pathways used by the most ancient bacteria and archaea, many of them involve reactions with hydrogen, carbon dioxide, and transition metal clusters like iron-sulfur complexes. These are exactly the conditions and catalysts available at alkaline hydrothermal vents. It's as if the chemistry of life retained a memory of its birthplace. Perhaps most tellingly, when scientists use genetic analysis to reconstruct the characteristics of Luca, the last universal common ancestor of all life on Earth, they find evidence of an organism that lived in just such an environment. Luca appears to have been anaerobic, unable to tolerate oxygen. It was autotrophic, making its own organic compounds from inorganic materials. Its metabolism was centered on reactions involving hydrogen, carbon dioxide, and iron-sulfur clusters, and it likely lived in a hot environment with abundant mineral catalysts. All of this points to hydrothermal vents, and particularly alkaline vents, as the, most likely, birthplace of life on Earth. But there's still a huge gap in this story. Even if we accept that alkaline vents provided the right environment and energy source, we still need to understand how non-living chemistry became living biology. We need to understand how molecules learned to replicate themselves, how they became enclosed in membranes, how they developed the ability to evolve. We need to understand, in other words, what the first living things were actually like. Chapter 4: The RNA World and the Molecule of Life. To understand the origin of life, we first need to understand what life is. That might sound obvious, but it's actually a profound question. What distinguishes living things from non-living matter? Modern life has certain hallmarks. It's cellular, bounded by membranes. It metabolizes, harvesting energy from the environment. It maintains homeostasis, keeping its internal conditions stable. It responds to stimuli. It grows and develops. But perhaps most fundamentally, life reproduces, creating copies of itself, and it evolves, changing over generations in response to environmental pressures. For life to reproduce and evolve, it needs two things: information and function. It needs a way to store instructions for building itself, and it needs the molecular machinery to carry out those instructions. In modern life, this division of labor is elegantly solved. DNA stores the information, acting as a stable genetic blueprint. RNA serves as a messenger, copying the information from DNA and carrying it to where it's needed. And proteins do most of the actual work, acting as catalysts for thousands of different chemical reactions, forming structures, transporting molecules, and defending against threats. But this system presents a chicken and egg problem for the origin of life. DNA can't function without proteins to copy it and maintain it. Proteins can't be made without DNA to encode their sequences and RNA to translate those sequences. And none of these molecules can replicate themselves without help from the others. They're all interdependent. So which came first? How could this complex system have arisen? For much of the 20th century, this seemed like an insurmountable paradox. Then, in the 1980s, came a discovery that would transform our understanding. RNA could do both jobs. The breakthrough came independently from two researchers, Thomas Cech and Sidney Altman, studying different biological systems. Cech was investigating how certain RNA molecules in a single-celled organism called tetrahemina were processed. He found that the RNA could splice itself, cutting out sections and rejoining the ends without any help from protein enzymes. The RNA was catalyzing its own modification. Altman, meanwhile, was studying an enzyme called ribonuclease P, which plays a role in processing other RNA molecules. He discovered that the active component of this enzyme, the part that actually performed the catalysis, wasn't the protein component as expected. It was the RNA. These were the first ribozymes, RNA molecules that act as catalysts. The discovery earned Czech and Altman the 1989 Nobel Prize in Chemistry and revolutionized thinking about life's origins. If RNA could act as both an information storage molecule and a catalyst, then perhaps RNA came first, before the evolution of DNA and proteins. This idea became known as the RNA world hypothesis. The proposal is that there was a stage in life's evolution when RNA performed all the key functions that in modern cells are divided among DNA, RNA, and proteins. RNA stored genetic information, RNA replicated itself, and RNA catalyzed the chemical reactions necessary for life. Support for the RNA world hypothesis comes from many directions. First, there's the fact that RNA still plays absolutely central roles in modern cells, roles that seem almost like evolutionary fossils from a more ancient time. The ribosome, the molecular machine that synthesizes all proteins in all organisms, is fundamentally an RNA structure. The protein components are important for stability, but the actual catalytic work of forming peptide bonds between amino acids is performed by ribosomal RNA. The ribosome is, in essence, a ribozyme. Moreover, many of the most fundamental cofactors in cellular metabolism, molecules like NAD+, FRD, and coenzyme A, all contain nucleotide components. It's as if modern metabolism still bears the signature of an earlier time when RNA and its close relatives were the primary functional molecules. Laboratory experiments have also shown that RNA has remarkable catalytic potential. By creating large libraries of random RNA sequences and selecting for specific functions, researchers have produced ribozymes that can perform an amazing array of chemical reactions. There are ribozymes that can catalyze carbon-carbon bond formation, that can copy other RNA sequences, that can join amino acids together, that can attach themselves to other molecules. While no one has yet created a ribozyme that can completely replicate itself, progress has been steady. And the goal doesn't seem impossibly far off. But the RNA world hypothesis also faces significant challenges. The first challenge is purely chemical. RNA is difficult to make from scratch. The 4 nucleotide building blocks of RNA, adenosine, guanosine, cytidine, and uridine, each require multiple steps to synthesize from simpler precursors. For decades, prebiotic chemists struggled to find plausible pathways that could produce these nucleotides under early Earth conditions. The ribose sugar that forms the backbone of RNA proved particularly problematic. When you try to synthesize ribose from simple starting materials like formaldehyde, you don't just get ribose. You get dozens of different sugars, many of which would interfere with RNA formation. How could the right sugar have been selected from this mess? Recent work has made progress on these problems. Chemist John Sutherland and his colleagues discovered pathways to nucleotide synthesis that avoid making ribose as a separate step. Instead, building the nucleotides through reactions that naturally favor the right products. These reactions work better in certain conditions. such as in the presence of phosphate minerals or under cycles of wetting and drying. While debates continue about exactly how prebiotic nucleotide synthesis occurred, it no longer seems like an impossible hurdle. A second challenge is concentration. Even if nucleotides formed on the early Earth, they would have been dilute, scattered across vast volumes of water. For RNA molecules to form, nucleotides need to link together, and this generally requires them to be close together in relatively high concentrations. This is where the alkaline vent hypothesis dovetails beautifully with the RNA world. The micropores in vent structures could concentrate molecules through various mechanisms. Temperature gradients in the pores could drive thermophoresis, causing molecules to accumulate in cooler regions. Cycles of wetting and drying, if parts of the vent system periodically dried out, could concentrate molecules dramatically. The mineral surfaces in the vents could adsorb molecules, holding them in place where they could interact. A third challenge is stability. RNA is a fragile molecule. The chemical bond that links the phosphate backbone can be broken by simple hydrolysis, especially in hot water. This instability seems at odds with the need for RNA molecules to persist long enough to replicate and evolve. However, some scientists argue that this very instability might have been advantageous in the early stages. A system where molecules are constantly being broken down and reformed might explore chemical space more rapidly than a system locked into stable but less functional molecules. Instability could have accelerated evolution. Only later, as RNA molecules evolved ribozymes that could modify and protect other RNAs, would longer-term stability become possible. The RNA world hypothesis doesn't propose that RNA-based life emerged de novo, fully formed. Rather, it envisions a gradual process. Simple organic molecules accumulated and reacted in favorable environments, perhaps in the micropores of alkaline hydrothermal vents. Random formation of short RNA-like polymers occasionally produced sequences with weak catalytic activity. Those molecules that happened to catalyze useful reactions, particularly reactions that helped produce more copies of themselves, would become more abundant. Over time, through countless cycles of random variation and selection, RNA molecules would become better and better at replicating themselves and at catalyzing useful chemical transformations. At some point in this process, a critical threshold was crossed. A set of RNA molecules emerged that could, collectively, reliably copy themselves and produce the molecular tools needed for their own maintenance. These molecules would constitute a primitive genetic system, capable of evolution in the Darwinian sense. This RNA world wouldn't have lasted forever. RNA's limitations, its instability, and its relatively limited catalytic repertoire compared to proteins, would have created selective pressure for improvements. The evolution of DNA, which is more stable than RNA thanks to a small chemical difference, would have provided better long-term information storage. The evolution of proteins, with their 20 different amino acid building blocks, offering far greater chemical diversity than RNA's 4 bases, would have provided more efficient and versatile catalysts. Perhaps proteins originally evolved as helpers for ribozymes, small peptides that bound to RNA and enhanced its catalytic activity. Perhaps DNA evolved as a more stable way to store the genetic information that RNA was passing from generation to generation. Whatever the exact sequence, the modern 3-part system of DNA, RNA, and proteins gradually emerged, each component taking on the role it performs best. And at some point in this transition, these self-replicating molecules became enclosed within membranes, creating the first true cells. This step was crucial because it allowed the genetic information and the products of that information to be kept together, creating discrete individuals that could compete and evolve as units. Chapter 5. The first membrane's life requires boundaries. Without some way to separate self from environment, there can be no distinct individuals, no way for beneficial innovations to be passed from parent to offspring, no way for natural selection to operate. The origin of cellular membranes was therefore one of the most critical steps in the emergence of life. But how did those first membranes form? What were they made of? And how could they have arisen through natural processes on the early Earth? Modern cell membranes are marvels of engineering. They're composed primarily of phospholipids, complex molecules with a phosphate-containing head group and two long hydrocarbon tails. These molecules spontaneously organize into bilayers, with the water-loving heads facing outward toward the aqueous environment, and the water-fearing tails facing inward, away from water. This bilayer forms the basic structure of all cell membranes, about 5 nanometers thick, flexible yet strong. Embedded in this lipid bilayer are thousands of different proteins. Some form channels that allow specific molecules to pass through the membrane. Others act as pumps, actively transporting materials against concentration gradients. Still others serve as receptors, sensing signals from the environment. The membrane is not a static barrier, but a dynamic, responsive interface. The sophistication of modern membranes poses an obvious problem for origin of life scenarios. Phospholipids are complex molecules that require multiple enzymatic steps to synthesize. The membrane proteins are even more complex. How could such elaborate structures have arisen before there was life to make them? The answer, most scientists believe, is that the first membranes were much simpler. They were probably composed not of phospholipids, but of fatty acids. Fatty acids are simpler molecules, essentially just a hydrocarbon chain with an acidic carboxyl group at one end. They're amphiphilic, meaning one end is water-loving and the other is water-fearing, just like phospholipids. And crucially, fatty acids can be made through purely abiotic chemistry. Studies of meteorites, particularly a class called carbonaceous chondrites, have shown that fatty acids form readily in space. The famous Murchison meteorite, which fell in Australia in 1969, was found to contain more than 70 different amino acids and a wide variety of other organic compounds, including fatty acids with chain lengths ranging from 2 to 8 carbon atoms. If meteorites brought these compounds to early Earth, or if similar chemistry occurred on Earth itself, fatty acids would have been available. When fatty acids are dissolved in water at sufficient concentration, they spontaneously form structures. At low concentrations, they form micelles, spherical arrangements with the fatty tails pointing inward and the heads facing the water. But at higher concentrations, and particularly when mixtures of different fatty acids are present, they form vesicles, hollow spheres bounded by a membrane. Unlike the bilayer membranes of phospholipids, fatty acid membranes can be either monolayers or bilayers, and they're much more dynamic. The individual fatty acid molecules are constantly exchanging between the membrane and the surrounding solution, between different areas of the membrane, and even between the inner and outer layers. This dynamism, which would be a liability for modern cells requiring tight control, over what enters and exits might have been an advantage for primitive protocells. Laboratory experiments have demonstrated that fatty acid vesicles can form under a wide range of conditions. Remarkably, they form more readily in the warm, alkaline, salty conditions found at hydrothermal vents than in cooler, fresher water. A study published in 2019 by Sean Jordan and Nick Lane showed that when a mixture of different fatty acids and fatty alcohols, another type of simple amphiphile, is placed in conditions mimicking a hydrothermal vent, stable vesicles form spontaneously. The mixture and the harsh conditions, which had been thought to be obstacles to membrane formation, actually promoted it. These protocell membranes have several properties that make them suitable for the early stages of life. First, they're permeable. While modern phospholipid membranes are excellent barriers that allow cells to carefully control their internal environment, they're almost too good. Small molecules, like nutrients, need to get in somehow. Modern cells solve this with elaborate protein channels, but protocells didn't have those. Fatty acid membranes, being less tightly packed, allow small molecules to pass through relatively freely. Nucleotides, amino acids, and other small organic molecules can enter protocells without specialized transport machinery. Second, they're responsive to their environment. When there's a difference in the concentration of small molecules across the membrane, fatty acid vesicles can grow by absorbing more fatty acids from the environment, or they can shrink. When they grow large enough, they can spontaneously divide, either through physical shear forces or through phase transitions in the membrane. This means protocells could have a primitive form of growth and division without requiring any elaborate cellular machinery. Third, they're compatible with RNA. Some early experiments suggested that RNA and fatty acid membranes might be incompatible, with charged RNA molecules disrupting membrane integrity. But more recent studies have shown that membranes composed of mixtures of fatty acids and fatty alcohols remain stable even in the presence of RNA and of divalent metal ions like magnesium, which RNA requires for its structure and catalytic activity. Experiments have even shown that protocells can exhibit competition and evolution. When vesicles containing RNA are mixed with vesicles not containing RNA, and the system is provided with more fatty acids, the RNA-containing vesicles grow faster. This happens because RNA inside the vesicle creates an osmotic imbalance, drawing in water and causing the membrane to expand, which in turn pulls in more fatty acids. The RNA, in essence, helps its vesicle compete for resources. If the RNA inside also has catalytic activity that produces molecules useful for the protocell, the advantage becomes even greater. A protocell containing a ribozyme that makes more fatty acids, or that produces molecules that help stabilize the membrane, would outcompete protocells without such useful RNA. Natural selection would operate at the protocell level, favoring vesicles that contained more functional genetic material. Research has even demonstrated a primitive form of heredity in such systems. When protocells grow and divide, the RNA inside is partitioned between the daughter cells. If those RNA molecules can replicate, perhaps through template-directed synthesis, using mineral catalysts or primitive ribozymes, then the genetic information can be propagated across generations. Mutations in the RNA sequence could lead to new functionalities, and beneficial mutations would spread through the population. This work is still in its early stages, and there are many gaps in our understanding. We don't yet have a complete protocell that can grow, divide, and evolve indefinitely using only plausible prebiotic chemistry. But the progress has been remarkable, and many scientists believe we're on the right track. One particularly fascinating recent discovery relates to how lipids might have been synthesized on the early Earth. A team led by Neil DeVarage at UC San Diego found that when a simple amino acid, cysteine, is mixed with certain short-chain thioester molecules in the presence of silica glass, they spontaneously react to form longer lipids that can assemble into vesicles. The silica acts as a catalyst, and critically, this reaction works at lower concentrations than would otherwise be necessary. Silica would have been abundant on early Earth, present in sand, clay, and the mineral structures of hydrothermal vents. The fact that it can catalyze lipid formation suggests a pathway by which the first membranes could have emerged from simple starting materials in geologically common environments. Other research has shown that the micropores in mineral structures, like those in hydrothermal vent chimneys, might have served as templates for the first protocells. Clay particles, particularly a type called montmorillonite, have been shown to promote both the formation of lipid vesicles and the polymerization of RNA. Some researchers have proposed that clay-armored bubbles with particles of clay stuck to the outside of a lipid membrane might have been an intermediate step between purely mineral compartments and free-living cells. All of this points to a picture where the emergence of cellular life wasn't a single sudden event, but a gradual process. Simple organic molecules accumulated in favorable environments. Amphiphilic molecules self-assembled into membrane-bounded compartments. RNA or RNA-like polymers formed inside some of these compartments, occasionally possessing catalytic activity. The compartments that happened to contain functional RNA were more successful at surviving and reproducing. Over many generations, the RNA became better at replicating and at performing useful chemistry, while the membranes became more sophisticated, perhaps through the incorporation of simple peptides that enhanced membrane stability or function. Eventually, this coevolution of genetics and structure reached a point where the protocells could be considered truly alive, capable of metabolism, growth, reproduction, and evolution. The exact moment when that threshold was crossed is probably impossible to define precisely. Life didn't appear all at once. It emerged gradually from increasingly complex chemistry. But emerge it did, somewhere around 4.2 to 4.3 billion years ago, likely in the mineral pores of alkaline hydrothermal vents, powered by natural proton gradients and catalyzed by iron sulfide minerals. Those first living cells would be almost unrecognizable to us. They had no true DNA, probably just RNA. They had no elaborate cellular machinery, just simple membranes and primitive ribozymes. They might not even have been free-living, remaining confined to the mineral structures where they arose. But from these humble beginnings, an entire living world would eventually evolve. Chapter 6. Luca, the last universal common ancestor. Every living thing on Earth today, from the smallest bacterium to the largest whale, from the humblest moss to the most complex human, shares a common ancestor. This isn't a religious belief or a philosophical conjecture. It's a scientific conclusion, supported by overwhelming evidence from genetics, biochemistry, and paleontology. We call this common ancestor Luca, the last universal common ancestor. Luca wasn't the first living thing. It wasn't even necessarily the only living thing at its time. But Luca is the organism from which all current life descends. Everything that came before Luca, all the other early life forms that must have existed, left no descendants. They went extinct, their lineages ending, while Luca's continued, branching and diversifying into the spectacular variety of life we see today. What was Luca like? When did it live? Where did it live? These questions have fascinated scientists for decades, and recent research has begun to provide some surprising answers. We can't observe Luca directly, of course. It lived billions of years ago, and cells don't fossilize well. But we can infer many of Luca's characteristics by looking at what all modern organisms have in common, and by using sophisticated genetic analysis to reconstruct what the ancestor of all life must have been like. The most comprehensive recent study of Luca was published in 2024 by a team led by Edmund Moody and colleagues at the University of Bristol. They analysed millions of genes from hundreds of bacterial and archaeal genomes, using computational methods to trace which genes were likely present in Luca and which evolved later. By comparing genetic sequences and accounting for horizontal gene transfer, where organisms swap genes, they built up a picture of Luca's biology. First, the timing. Luca lived approximately 4.2 billion years ago, with a confidence interval ranging from about 4.1 to 4.3 billion years ago. This means Luca existed just 400 million years after Earth formed, and just 200 to 300 million years after the end of the heavy bombardment. Life didn't wait long, in geological terms, to get started. Second, the environment. All the genetic evidence points to Luca being a thermophile, or possibly even a hyperthermophile, an organism that thrives at high temperatures. Many of the proteins that can be traced back to Luca show characteristics consistent with function at high temperatures. This suggests Luca lived in a hot environment, perhaps in or around hydrothermal vents. More specifically, Luca appears to have been adapted to an environment rich in hydrogen, carbon dioxide, and transition metals, particularly iron and nickel. This is exactly the kind of environment found at alkaline hydrothermal vents. The metabolic pathways that seem to be most ancient, present in both bacteria and archaea, and therefore likely inherited from Luca, are consistent with a lifestyle based on reacting hydrogen with carbon dioxide to produce organic compounds, using iron-sulfur clusters as catalysts. Luca was almost certainly anaerobic. It lived in a world before oxygen was abundant, and in fact, many of the enzymes that can be traced to Luca are damaged by exposure to oxygen. Luca would have found our modern, oxygen-rich atmosphere toxic. Luca was also autotrophic, meaning it made its own organic compounds from inorganic materials rather than consuming organic matter made by other organisms. This makes sense. If Luca is the ancestor of all life, it couldn't have depended on other organisms for food. The specific metabolic pathway that Luca seems to have used is called the Wood-Ljungdahl pathway, or the acetyl-CoA pathway, a series of chemical reactions that combines carbon dioxide and hydrogen to make organic compounds, particularly acetate. This pathway is still found today in certain bacteria and archaea, particularly methanogens, organisms that produce methane, and acetogens, organisms that produce acetate. The fact that both of these very different groups use this pathway suggests it's ancient, inherited from their common ancestor. And interestingly, this pathway requires exactly the kind of iron-sulfur clusters and conditions found at alkaline hydrothermal vents. Luca almost certainly had a proper genetic code, All organisms today use the same basic genetic code, where sequences of 3 nucleotides, codons, specify particular amino acids. This code is essentially universal, with only minor variations in a few organisms. The simplest explanation for this universality is that the code was already established in Luca and has been inherited by all its descendants. Luca had ribosomes, the molecular machines that translate genetic information into proteins. The ribosome is one of the most conserved structures in all of biology. Bacterial ribosomes and archaeal ribosomes have slightly different structures, but they're fundamentally similar, and certain parts are almost identical. This suggests the ribosome was already present and functional in Luca. Intriguingly, recent research suggests Luca may have even had primitive immune systems. The study by Moody and colleagues found genes in Luca related to defense against viruses. This implies that by 4.2 billion years ago, not only did cellular life exist, but so did viruses that infected those cells, and the cells had evolved mechanisms to fight back. Life was already engaged in evolutionary arms races. Luca was probably not free-living in the modern sense. The most widely accepted scenario is that Luca, and perhaps several generations of its ancestors, remained confined to the mineral structures of hydrothermal vents. The cells might have been little more than compartments within the micropores of the vent walls, using the natural proton gradients provided by the vent environment to power their metabolism. At some point, Luca's descendants evolved the ability to create their own proton gradients, rather than relying on those provided by the environment. This was a crucial innovation. Proteins evolved that could pump protons across membranes, creating an artificial proton gradient using energy from metabolism. Other proteins evolved that could use these gradients to synthesize ATP. With this innovation, cells were no longer tethered to hydrothermal vents. They could venture out into the open ocean, into new environments, and still maintain the energy supply they needed. This liberation from the vent environment would have opened up vast new opportunities for evolution. Not long after Luca, the tree of life split into its 2 main branches, bacteria and archaea. These are the 2 domains of prokaryotic life, single-celled organisms without a nucleus or other complex internal structures. For billions of years, prokaryotes were the only form of life on Earth. Much later, perhaps around 2 billion years ago, a momentous event occurred. One archaeal cell engulfed a bacterium, but instead of digesting it, the 2 formed a symbiotic relationship. The bacterium became the mitochondrion, the powerhouse of the cell, while the archaeal host evolved into the ancestor of all eukaryotes, organisms with nuclei, including plants, animals, fungi, and many single-celled organisms. But that's getting ahead of our story. In the time between Luca and the origin of eukaryotes, prokaryotic life would transform Earth in profound ways. Most significantly, some bacteria would evolve the ability to perform oxygenic photosynthesis, splitting water molecules to obtain electrons and releasing oxygen as a waste product. This seemingly simple metabolic innovation would change everything. For billions of years, Earth's atmosphere had little free oxygen. But as photosynthetic bacteria, particularly cyanobacteria, spread across the planet's oceans, they pumped out oxygen. At first, this oxygen reacted with dissolved iron in the oceans, precipitating out as massive rust deposits, the banded iron formations we can see in ancient rocks today. But eventually, around 2.4 billion years ago, the oxygen began accumulating in the atmosphere. This great oxidation event was catastrophic for many organisms. Oxygen is a highly reactive gas, and for life forms adapted to an oxygen-free world, it was poison. Many lineages went extinct, but other organisms adapted, evolving ways to cope with oxygen and eventually to use it. Aerobic respiration, using oxygen to extract maximum energy from food, became possible. This innovation would eventually lead to the evolution of large, complex, multicellular organisms like ourselves. Chapter 7. The Rock Record of Early Life. How do we know any of this really happened? How can scientists be so confident about events that occurred billions of years ago, when there were no witnesses and when most of the rock record has been destroyed by geological processes? The answer is that life left traces, faint but unmistakable, preserved in some of the oldest rocks on Earth. These traces come in several forms, and each tells part of the story of life's early history. The most direct evidence would be fossils, actual preserved remains of ancient organisms. But fossilization requires special conditions, and the older the rocks, the more they've been heated, compressed, and chemically altered by geological processes. Most ancient rocks have been transformed beyond recognition. The few that survive are precious windows into the deep past. The oldest rocks on Earth come from a few locations: the Jack Hills region in Western Australia, parts of Greenland, and areas of Canada. The Jack Hills contain zircon crystals, incredibly durable minerals that have survived for over 4 billion years. These zircons contain chemical signatures indicating they formed in the presence of liquid water, confirming that Earth had oceans by about 4.4 billion years ago. But zircons don't contain fossils. For evidence of life, we need sedimentary rocks, the kind formed by the accumulation of material at Earth's surface. And very few sedimentary rocks have survived from the early Archean eon. The oldest claimed evidence of life comes from rocks in the Nuvvuagittuq belt in Quebec, Canada. In 2017, researchers reported finding tiny tubes and filaments in these rocks that they interpreted as fossils of bacteria that lived near hydrothermal vents on the ancient seafloor. If confirmed, these fossils would be around 4.3 billion years old, making them the oldest evidence of life on Earth. However, this claim remains controversial. The structures are small and ambiguous, and some scientists argue they could have been formed by non-biological processes. When you're looking at rocks that have been transformed by heat and pressure for over 4 billion years, distinguishing biological from non-biological structures is extremely difficult. Less controversial are the stromatolites found in rocks from Greenland. In 2016, Alan Nutman and colleagues reported finding layered structures in 3.7 billion-year-old rocks that bear the hallmarks of stromatolites, formations created by mats of photosynthetic bacteria. Modern stromatolites still form today in a few places, like Shark Bay in Australia. They're created when films of bacteria, particularly cyanobacteria, grow on seafloors or lake beds. The bacteria trap and bind sediment particles, and they also precipitate minerals through their metabolic activities. As new layers of bacteria grow on top of old ones, distinctive layered structures build up, sometimes growing into columns or mounds. The Greenland stromatolites show these characteristic layered structures, along with other features consistent with biological origin, such as variations in layer thickness that might correspond to seasonal or tidal cycles. The rocks also contain organic carbon with isotopic signatures consistent with biological processing. Even more compelling are the 3.5 billion-year-old stromatolites from the Pilbara region of Western Australia. These structures are beautifully preserved, showing clear evidence of biological origin. They occur in rocks that were formed in shallow marine environments, just the kind of place where photosynthetic bacteria would thrive. Actual fossilized cells have been reported from rocks of similar age. The apex chert in Western Australia, about 3.5 billion years old, contains microscopic structures that look like simple filamentous bacteria. When first discovered by William Schopf in the 1990s, these were hailed as definitive evidence of early life. However, later research raised questions about whether some of these structures might be abiological, formed by mineral processes rather than biology. The debate over these very early microfossils remains intense. The problem is that bacteria are simple. A single cell is just a blob, and under a microscope, it can be hard to distinguish a fossil bacterium from a bubble or a mineral crystal. Scientists have developed multiple criteria to help make this determination: the presence of organic carbon, the right size and shape, evidence of cell division, presence in assemblages showing variation, and more. But ambiguous cases remain. Perhaps the most robust evidence for early life doesn't come from actual fossils, but from chemistry. Living organisms preferentially use the lighter isotope of carbon, carbon-12, over the heavier carbon-13. This is because the lighter isotope reacts slightly faster in metabolic processes. As a result, biological material tends to be enriched in carbon-12 compared to non-biological carbon sources. When scientists analyze the carbon isotope ratios in ancient rocks, they can look for this biological signature. Organic carbon that's been processed by life will have a distinctive ratio of carbon-12 to carbon-13, different from carbon that's never been part of a living organism. Such signatures have been found in rocks from Greenland that are 3.8 billion years old, and possibly in even older rocks. While carbon isotope ratios alone aren't definitive proof of life, some geological processes can produce similar patterns. When found in conjunction with other evidence like stromatolites or microfossils, they build a compelling case. By 3.5 billion years ago, the evidence for life becomes overwhelming. Multiple sites around the world contain stromatolites, microfossils, and isotopic signatures, all pointing to the presence of abundant microbial life. These early organisms were almost certainly photosynthetic, living in shallow marine environments where they could access sunlight. This means that by 3.5 billion years ago, life had not only originated, but had evolved considerably. The evolution of photosynthesis, particularly oxygenic photosynthesis, is a complex innovation requiring sophisticated biochemical machinery. The fact that it had already appeared by this time suggests life must have originated considerably earlier, giving evolution time to develop these capabilities. This timeline fits well with the genetic evidence from Luca. If Luca lived around 4.2 billion years ago, that gives evolution about 700 million years to go from the first living cells to the diverse, photosynthetic, ecosystem-building organisms we see evidence of by 3.5 billion years ago. That's a long time, but on the timescale of evolution, it's not implausibly long. What about the gap between Luca at 4.2 billion years ago and the Greenland stromatolites at 3.7 billion years? Where's the evidence from that half-billion-year window? The unfortunate answer is that most rocks from that period have been destroyed. Plate tectonics constantly recycles Earth's crust, subducting old oceanic crust back into the mantle where it melts. Continental rocks can survive longer, but even they get eroded, metamorphosed, and reworked. The handful of rocks we have from the early Archean are incredible survivors, but they're a tiny, fragmentary sample of what once existed. Still, what we can see tells a consistent story. Life originated on Earth very early, within the first few 100 million years after the planet became habitable. It started in extreme environments, likely hydrothermal vents, where chemistry was rich and energy was available. It spread and diversified rapidly, colonizing different environments and evolving new metabolic capabilities. By 3.5 billion years ago, microbial life covered the planet, living in shallow seas, in deep oceans, in hot springs, and probably many other environments we haven't yet found evidence for. Chapter 8. The Big Picture. Let's step back and look at the full sweep of what we've learned. The origin of life wasn't a single moment or event. It was a process, one that unfolded over hundreds of millions of years and countless intermediate steps. It began with chemistry. In the extreme environment of the early Earth, in places where conditions were just right, likely the micropores of alkaline hydrothermal vents, simple organic molecules accumulated. Amino acids, nucleotides, fatty acids, and countless other compounds formed through reactions powered by lightning, UV radiation, and geochemical energy. Some of these molecules had special properties. Certain RNA sequences, or RNA-like polymers, could catalyze chemical reactions. Some reactions were self-reinforcing, producing molecules that helped make more of themselves. Through endless rounds of random synthesis and selective preservation, molecular systems became more sophisticated. Amphiphilic molecules, fatty acids, and their relatives spontaneously assembled into membrane-bounded vesicles, creating compartments that could concentrate chemicals and protect them from dilution in the vast ocean. When genetic molecules became enclosed in these protocells, a crucial threshold was crossed. Now, variation could be heritable, and natural selection could operate on distinct individuals. The first living cells were probably not free-living, but confined to mineral structures, using natural proton gradients for energy. Their genomes were small, likely just RNA. Their metabolism was simple, probably based on reacting hydrogen with carbon dioxide to make organic compounds. But they could replicate, they could mutate, and they could evolve. Over millions of generations, these cells became more sophisticated. DNA evolved as a more stable information storage medium. Proteins evolved as more versatile catalysts. The genetic code became established. Ribosomes developed. Cells evolved the ability to create their own proton gradients, liberating them from dependence on hydrothermal vents. By around 4.2 billion years ago, there existed a cell sophisticated enough to be the ancestor of all subsequent life. We call it LUCA. From LUCA, the tree of life branched into bacteria and archaea. These two domains of prokaryotic life spread across the planet, evolving countless metabolic innovations. One of the most important innovations was photosynthesis, the ability to harvest energy from sunlight. Early photosynthesis probably didn't produce oxygen, using other electron donors like hydrogen sulfide or ferrous iron instead. But eventually, cyanobacteria evolved oxygenic photosynthesis, splitting water to obtain electrons and releasing oxygen as a byproduct. This oxygen would accumulate over billions of years, eventually transforming Earth's atmosphere and enabling new forms of life. The evolution of eukaryotic cells, through the symbiotic merger of bacteria and archaea, created a new domain of life with unprecedented complexity. And eventually, eukaryotes would evolve into multicellular organisms, including all the plants and animals we're familiar with. But that entire later history, all 4 billion years from Luca to today, rests on that initial chemical transition from non-life to life. Understanding that transition has been one of the great scientific quests of the past century. We don't have all the answers yet. We don't know exactly where life originated, though alkaline hydrothermal vents seem like the best candidate. We don't know the exact sequence of chemical steps that led from simple organics to self-replicating systems. We don't know how the genetic code became established, or how the ribosome evolved, or how cells first learned to perform photosynthesis. But the outlines of the story are becoming clear. The discovery of deep-sea vents and the recognition of their potential importance revolutionized the field. The RNA world hypothesis provided a plausible solution to the chicken and egg problem of genetic information versus catalytic function. Laboratory experiments have demonstrated that protocells can form, grow, and exhibit evolution under conditions that might have existed on early Earth. Genetic analysis has revealed the likely characteristics of Luxi and pushed back the timing of life's origin to astonishingly early in Earth's history. Perhaps most importantly, we've learned that life's origin wasn't miraculous or inexplicable. It was the result of chemistry operating under the right conditions. The molecules of life are complex, but they're not impossibly so. The pathways from simple to complex can be traced, even if many details remain uncertain. This has profound implications for the question of life elsewhere in the universe. If life arose on Earth as soon as conditions permitted, and if it arose through ordinary chemical processes operating in environments that aren't particularly unusual, then life might be common in the universe. Any planet with liquid water, sources of chemical energy, and a few 100 million years of stable conditions might see the emergence of life. The universe contains hundreds of billions of galaxies, each with hundreds of billions of stars. Many of those stars have planets. We're discovering thousands of exoplanets, and many are in their star's habitable zone, where liquid water could exist. Some of Jupiter's and Saturn's moons have subsurface oceans that might harbour hydrothermal vents, not unlike those where life may have begun on Earth. If life emerged elsewhere through similar processes, we might expect it to share certain characteristics with Earth life. It would need some form of information storage and replication. It would need catalysts to speed up chemical reactions. It would need compartmentalization to create distinct individuals. It would need to harness energy from its environment. The specific molecules might be different. The details might vary, but the fundamental principles might be universal. Or perhaps not. Perhaps life on Earth took advantage of uniquely favorable circumstances and represents a rare accident in a mostly lifeless cosmos. Perhaps the gap between complex chemistry and true life is wider than we think, and Earth just happened to bridge it through extraordinary luck. We don't know yet, but we're looking. Missions to Mars search for evidence of past microbial life. Missions to Europa and Enceladus will investigate the subsurface oceans of Jupiter's and Saturn's moons. The James Webb Space Telescope and future instruments will analyze the atmospheres of exoplanets, searching for biosignatures, combinations of gases that might indicate life. In the coming decades, we may find out whether we're alone. And if we find life elsewhere, the first question we'll ask is, how did it begin? Is its origin story similar to ours? Or did it find a completely different path from chemistry to biology? Chapter 9. The Continuing Mystery. As we come to the end of our journey through the origin of life, it's worth reflecting on what an extraordinary story this is. 4.5 billion years ago, there was no life anywhere on Earth. Just 400 million years later, maybe less, there was Luca, a sophisticated cell with a genetic code, metabolism, and the ability to reproduce and evolve. Today, billions of years later, Earth teems with millions of species, from bacteria to blue whales, from roses to redwoods, from fungi to humans. All of it traces back to that first spark, to those first molecules that learned the trick of making copies of themselves, to those first cells that grew and divided in the mineral labyrinths of hydrothermal vents. We're all the descendants of Luca, all built from the same basic biochemical toolkit, all using the same genetic code, all playing out variations on strategies that worked 4 billion years ago in a world nothing like our own. The story of life's origin is still being written. Every year brings new discoveries, new experiments, new insights. Perhaps someday we'll understand it completely, able to write out every chemical step from simple molecules to living cells. Perhaps we'll even recreate the process in the laboratory, building artificial life from scratch and demonstrating that the emergence of life is, given the right conditions, not just possible, but probable. Or perhaps the origin of life will always retain some mystery, some elements that are lost to the depths of time and can never be fully recovered. Either way, the quest to understand our ultimate origins is one of the most profound and important endeavors in all of science. Because in understanding where we came from, we understand what we are. We're not separate from nature, not above or outside the natural world. We're part of it, products of the same chemical and evolutionary processes that gave rise to all life. The atoms in our bodies were forged in dying stars billions of years ago. The molecules in our cells follow patterns established in Earth's earliest life forms. The energy that powers our thoughts is generated by molecular machines inherited from ancient bacteria that became our mitochondria. We are, in a very real sense, the universe become conscious of itself. We're made of star stuff, as Carl Sagan said, arranged in ways that allow us to wonder about our origins, to ask questions, to seek answers. And on this warm evening, as you drift towards sleep, you can take comfort in knowing you're part of this ancient lineage. Every cell in your body carries echoes of that first spark 4 billion years ago. You're connected to every living thing on Earth through an unbroken chain of ancestors stretching back to Luca and beyond. In the morning, when you wake, life will continue. Bacteria will be reproducing. Plants will be photosynthesizing. Animals will be hunting and eating and growing. The grand experiment that began in those hydrothermal vents so long ago will continue, exploring new possibilities, adapting to new challenges, creating new wonders. And somewhere, perhaps on a distant world, orbiting another star, chemistry might be crossing that same threshold right now. Other molecules might be learning to replicate. Other protocells might be competing and evolving. Other forms of life might be taking their first tentative steps toward complexity, beginning their own 4 billion year journey toward who knows what. The origin of life is not just ancient history. It's a process that might be happening elsewhere right now. And it's a mystery that illuminates the very nature of existence. Thank you for joining us on this journey through time and chemistry and the emergence of living things. If you found this exploration calming and thought-provoking, please consider liking this video and subscribing to Bedtime Biology. We'll continue bringing you more quiet explorations of life's wonders, one relaxing night at a time. Sleep well, and may your dreams be filled with ancient oceans, bubbling vents, and the first stirrings of life.